Thank you.
Good day, everyone.
Good day.
Good
Hello.
day. Yeah. Good day. Thank you for joining. Good morning, good afternoon, good evening, everyone, wherever you're joining from. Thank you for joining the August edition of the GET webinar series. Thank you. We are so glad to have you. And to our distinguished speakers, our colleagues, our partners, researchers, policymakers, students, and participants from all over the world. We are glad to have you here. And our speakers, we have quite a lineup of experts with us. We have Ms. Severance, we have Dr. James, we have Dr. David, we have Dr. Trot, and Dr. Mary. Thank you so much for joining us. We are glad to have you. And on behalf of the Global Emerging Pathogens Treatment Consortium, GET, it is my pleasure to welcome you once again to the August edition of the GET Webinar Series 2026. The Chief Operating Officer, Dr. Ayodotun Babadoye, sent his regards, and my name is Faith Omalade Adebayo. I'm a Research and Project Officer at GET Consortium, and I am delighted to serve as your moderator for today's session. And again, we sincerely appreciate everyone who has taken their time to join us today, particularly to our speakers. We are especially grateful for accepting our invitation and for making time to join and contribute your expertise to this important conversation. We are honored to have you with us and bringing perspective from international biosecurity policy, biotechnology industry, public health, biosafety, regulatory governance, and defense science. Thank you so much. Briefly, before we start properly, allow me to briefly introduce GET Consortium. The Global Emerging Pathogens Treatment Consortium, GET, was established in 2014 in response to the devastating Ebola virus disease outbreaks in West Africa between 2014 and 2016. The consortium works to develop African-led and Afrocentric strategies for addressing emerging biosecurity and global health threats within a broader international context. We collaborate with organizations, institutions, experts, researchers, policymakers, and other stakeholders both within and outside Africa. And our work spans areas including capacity building, research, logistics, publications, community engagement, policy advocacy, strengthening preparedness against emerging and also re-emerging health threats. And this webinar series is a platform for experts and stakeholders to exchange knowledge, examine emerging challenges, and also explore practical solutions to some of the most important issues that we are facing globally and in health security. So today's conversation comes at a very particularly important time, and we understand that biotechnology is advancing rapidly and changing what is possible in health, scientific research, agriculture, industry, and even sustainable development. And technologies such as genomic sequencing, gene synthesis, advanced biological research, scientific biology, and other emerging life science technologies are opening new opportunities for addressing some of the world's most pressing challenges. And for Africa, this development presents an enormous opportunity for us. They can contribute to improved disease surveillance, diagnostics, vaccine and therapeutic development, agricultural productivity, research capacity, and a broader economic and scientific development. However, this same technology that creates opportunity can also introduce new challenges, which is why we have to be prepared for it. And as these biological capacities become more sophisticated and increasingly accessible, so there are questions around biosafety, biosecurity, dual use research, ethics, and responsible innovation, governance, and preparedness that's become increasingly more important. So the challenge before us now, therefore, is to simplify how to simply describe and talk about how to accelerate biotechnology and to ensure that biotechnology advances responsibly, safely, ethically, and in ways that can maximize the benefits while reducing potential risk, which is why this conversation is particularly important for Africa. And today's webinar is themed Biotechnology, Biosecurity, and Africa's Future, balancing innovation with responsible governance. Today, we'll be examining issues from several complementary perspectives, from global biosecurity governance to industry standards to laboratory biosafety to responsible biotechnology regulation and governance of dual-use research. Without wasting more of our time, before we proceed, I would also like to mention a few housekeeping rules. Please introduce yourself in the chat box. Let us know wherever you're joining us from. For example, my name is Faith. I am joining from Nigeria. And if you have questions, please direct your questions to the Q&A session. Do not post your questions in the chat box so it doesn't get lost in chat. We will be able to trace it during the question and answer session. And we also encourage you to remain engaged throughout the session. This webinar is also being recorded and it will be posted on our YouTube channel at Get Africa after the webinar. Thank you very much. And now, moving on to our first speaker. To begin our discussion, it is my pleasure to introduce our very first speaker for today's discussion, Ms. Severance, who is the Deputy Vice President at the Nuclear Threat Initiative, NTI, where she leads efforts to reduce biological risk to humanity. She joined NTI in 2018 and previously served in the U.S. Department of Defense, supporting the Cooperative Threat Reduction Program and the Global Health Security Agenda. She has extensive experience in biosafety, biosecurity, biological threat surveillance, detection, and response. Ms. Severance holds an MPH in infectious disease epidemiology from George Washington University and a BS in public health from Rutgers University. She brings expertise from global biosecurity policy space, and her work is closely connected to international efforts to strengthen preparedness, reduce biological risk, and promote responsible approaches to biotechnology governance. Today, she'll be speaking on strengthening global biosecurity, current efforts, and priorities for responsible biotechnology governance. Ms. Severance, we are delighted to have you with us today. Thank you so much once again for accepting our invitation. The floor is yours.
Well, thank you very much, Faith, and thank you to Get for inviting me here today and joining this very timely and important conversation. So as Faith mentioned, Haley Severance, and Deputy Vice President at NTI's Biological Policy and Programs team. And I'm gonna pull up my slides while I'm talking, and you can let me know if you can see them. Okay. All right, I see a thumbs up. Thank you. So, I mean, Faith set the scene beautifully, so I don't think I have much work to do there. But, you know, a little bit of detail that shows us just how quickly this space is moving. You know, traditional protein binder design, for example, used to take years of iterative lab work. Today, AI tools can generate thousands of candidate binders computationally in days, narrowing really that field down to a much smaller set before you even reach bench research. And, you know, that's extraordinary news for vaccine and treatment development. You know, a better understanding and use of protein binders, for example, in therapeutics can really help deliver cancer treatments to the cancerous cell rather than harming those non-cancerous cells around it, just as an example. But these AI capabilities that let us engineer living systems, which I'll refer to as AI bio capabilities, really cut both ways. They can help us detect the next outbreak faster. They can help us design the countermeasure sooner. But in the wrong hands and without the right safeguards, they can make it easier to cause deliberate or accidental harm. And that risk is really growing faster than our ability to govern or manage it. And at NTI Bio, we see our job as helping to close that gap between fast-moving technology and slower-moving governance. How do we get that gap smaller? So I'm going to walk you through two efforts that we have underway. One that's really more kind of globally facing, and one that's focused specifically in Africa and on Africa and what each is telling us. So before I go much further, sorry, I'm trying to figure out how to fit. Okay, a little bit about who we are at NTI Bio. NTI is a nonprofit, nonpartisan organization located in Washington, D.C., and we focus on reducing nuclear, biological, and emerging technology risks imperiling humanity. Biological threats we know, whether they are natural, accidental, or deliberate, can kill millions, cost billions, and destabilize countries and economies, well beyond where they may start. And our mission is to reduce those catastrophic biological risks. And in order to do this mission, we really work with governments, industry, academia, and international organizations to test solutions and advocate for those systemic changes that this problem set requires. And so one of the solutions that we've started to try to address this issue set is our AI Bio Global Forum. We really kind of saw this gap coming. It's not a new story that technologies move faster than our ability to govern them. And we really wanted a place where the people building these tools and capabilities can talk with those who have a responsibility to govern them. And the AI Bio Forum is what we saw as meeting that need. It's an international convening that brings together AI developers, biosecurity experts, policymakers, all working to identify and reduce biosecurity risks that come at that intersection of AI and the life sciences. And we built it around these three goals that you see on the slide, really developing a shared understanding of the risks, supporting tools and practices that protect good uses while guarding against, potential misuse, and pushing for adoption of national and global governance mechanisms. And so I'll walk through kind of what we've accomplished and where we're going with this forum. So in July of last year, more than 35 leading experts came together to issue a joint statement drafted through the forum on the biosecurity risks at this AI life science convergence. Both the ways that these tools lower the barrier for someone trying to cause harm, the ways they raise the ceiling on how much harm is possible, and how these capabilities are reducing the effectiveness of currently available biosecurity and biodefense measures. The signatories include, luminaries in this field such as Yoshua Bengio, former heads of state Helen Clark, researchers like Andrew Yao, George Church, David Relman, and many others. But this wasn't just a Western document. We had representatives from Nigeria, India, Korea, Singapore, China, and beyond join on this statement. And we took this statement to the BWC working group session in August 2025, where GET was represented on our panel. And we've been using it ever since to reach out to national policymakers and governments to, show them the risks that we're concerned about and begin to discuss how to address them. And we've, this wasn't kind of like a one-time thing. We've kept the momentum going through the forum. And I'd like to talk now about some activities we have ongoing in the AI Bio Global Forum. One of these activities is what we call our horizon scanning process. And basically every 6 months or so, which seems like an eternity in AI time, but we ask, a pretty simple question. Is governance keeping pace with what these tools can now do? And this is done through our Horizon Scanning Risk Assessment and Evaluation Working Group within the forum. And we track capability advances against existing guardrails. And these products, again, which we put out regularly, are designed to help policymakers, industry, and researchers anticipate, what's coming and what tools we have, potentially to govern it. The spring scan, for example, found that, we're making steady incremental progress. There's tighter integration between LLMs and biological research tools, more automation and lab integration, and real advances in protein and genome models, but the governance is really lagging behind all of it. And, that's the report, the purpose of this report, to give people early insight they need to make stronger safety and security decisions before that gap widens further. So that's one example of our work through the forum. Another is kind of more on so that was more on the risk assessment side, and this is more on the developing safeguards to address that risk side. So under our biological AI models working group, we're advancing some efforts that are co-developed with international advisors. And the first that we put out in January of this year is a framework for managed access. And, the instinct, in this space, and I've read a lot of articles about this, is framing things as open versus closed. And we think that's, a false dichotomy. Managed access is about balancing risk reduction with equitable access, not choosing, one over the other. And we built a framework to help provide ideas for how to do this. And we built it on more than 20 interviews with experts in biosecurity, in biological AI tool development and research. And, it rests really kind of on two main principles. One, that access should be tiered based on a tool's risk level, and that the need for security has to be balanced against the need for equitable access. And these, principles shape how we actually developed the framework. And you see, for example, here, some ideas about how to tier access based on that risk level. And we also, laid out what it takes to put managed access into practice, for example, through platforms, the infrastructure that may be needed, governance approaches, and how to weigh in equity considerations. So, moving forward, these platforms are going to be essential to whether this framework actually works in the real world and not, just on paper. And we see some examples, where, for example, managed access is being put into place to guard against very highly capable LLMs and chatbots in the biological space. Another effort, again, through our biological AI model working group, is something we call the metadata standard. And sorry, this slide looks complicated, but let me kind of explain the simple idea behind it. The idea is that every biological design should carry its own data regarding how that design came about, its provenance. So the standard helps track the origin of a sequence that's developed through use of a biological AI tool, including every edit or operation that was performed as it moves, for example, between tools. And each tool adds its own layer of information as a design passes through it. And that metadata file travels with the sequence and is linked to it with a checksum, so the two can't be separated. And by the time an order, for example, a sequence order reaches a DNA synthesis provider, they're not only looking at the sequence that may or may not have homology to anything, currently known. They have the context, where this design came from, what's been done to it, why does it look the way it looks? And so we're piloting this with industry now. I see James is speaking after me, and he's been really helpful in this effort. And the goal is, accountability without slowing down the science. Another example of work we're doing through the forum is something that we think addresses a really important gap. Frontier LLM developers are building safeguards into their models, through ongoing evaluation. They're building in managed access controls. And, these may not be perfect. In fact, they're definitely not perfect, especially for biology, but they exist. But a huge gap is that biological AI models really don't have any of those safeguards. And so we've built a proof of concept to start closing that gap, focused on AI tools that design proteins which bind to other proteins important to human health. So those protein binders that I mentioned at the beginning. And most proposed safeguards work by matching a sequence against a list of known dangerous ones. And that may not completely work here because AI can design a protein that's never existed before, and there's nothing to kind of like match it against. What we're proposing we screen for is what the user asks for. So before the model generates anything. So we look at the target that they're trying to, match with, and the biological structure the user wants a protein designed to bind to. So we do that using a protein language model that's learned to recognize what a protein does and how it's shaped, not just, the sequence. And because two proteins could look totally similar on paper and function almost identically, or the reverse, so a sequence isn't, completely enough. And we're evaluating that target again before the novel design exists, rather than trying to assess something brand new after the fact. And so it's an easier problem we're trying to solve. And this is, an early step to figuring this out. It needs refinement, especially kind of the underlying database of what we deem as harmful targets, or that should be flagged. But, it's a first step, and we think it'll be most powerful when paired with, the trusted user access programs, for example, like what CEPI is already piloting. So a few things that, so I kind of summarize a few things that are coming out of the working groups from the forum, and how we've really kind of like sharpened how we think about these issue sets, and are coming up with, some real promising solutions. But we're constantly coming across, new issue sets as the technology evolves. And we really appreciate help on this. We want more members to join the forum, because more knowledge helps us develop better solutions. So if you want to help us with this effort, please, check out the website, and there are details there about the forum. And so, that's kind of a summary of some of the things we're doing kind of more on the global scale. And so now let me shift to what this push toward responsibility amid advances in technology looks like, on the ground, more in the African context. So, sorry, I had an animation, hopefully. Okay. So in July, we released our new Africa Health Security Index, which is a look at epidemic and pandemic preparedness across all 54 African countries that are member states to the international health regulations. And we built this index with Brown University Pandemic Center, Economist Enterprise, and Science for Africa Foundation. We built it on the same open source methodology as the Global Health Security Index, but we didn't just completely port that framework over. We convened a reference group of African experts and institutions to make sure it actually reflected African priorities and realities, not assumptions that were developed elsewhere. So working with the reference group, we really kind of dug into these 6 categories listed on this slide. And I'll note that the reference group identified biosafety and biosecurity as a priority they really wanted to address through this Africa edition of the Health Security Index. And before I move on to kind of like more details in the biosafety and biosecurity section, I will say, the overall finding from the Africa Health Security Index is that health security capacity is genuinely and measurably growing across the continent, but significant gaps remain. And so let me move to the biosafety and biosecurity portion, and I'll provide some more details on this. So these, of course, are the capacities that help prevent both accidental and deliberate biological events. And these scores really have improved since 2021, but they're still pretty low. All of these scores are on a scale of 0 to 100. So a 14.9 for biosecurity was the continental average, and 18.2 for biosafety, and just 1.9 out of 100 for dual use research of concern. So again, this is an improvement from 2021, but still quite a bit of room to grow. We do see quite significant growth, for example, in national laboratory capacities. 76% of countries now have national laboratory subject to external quality assurance review, up from 58% in 2021. 56% now have nationwide specimen transport systems compared to just 19% 5 years ago. But as laboratory capacity grows this fast, the biosafety and biosecurity investment really needs to grow alongside it. And so how do we build systems that are more capable and more secure at the same time? And we've really built this index to be used by governments, regional bodies, funders, multinational organizations, and the private sector. And it comes with recommendations for where to act first. National leaders can use it to guide preparedness investment. International and regional organizations can use it to track progress and make case for stronger commitments. There are 29 recommendations in total, and they're all available on the website. I will not walk through them here, but they are organized by focus area and by who needs to act upon them. So I'll walk through a couple of the relevant ones to this conversation. So these are the recommendations that really are focused on how do we strengthen biosafety, biosecurity, and responsible biotechnology governance. The first is requiring DNA synthesis screening at the point of procurement. So at least 7 African countries already have an active gene synthesis market through third-party vendors or domestic providers, but not one has legislation requiring that synthesis orders be screened. So if you require researchers to buy from companies that screen, you create a market incentive for responsible producers. And that really extends the benefits of these policies even beyond, African countries' borders. The second recommendation is around developing dual-use research of concern oversight frameworks at the national level, with guidance from the continental regional level. Only 6 countries currently have a designated agency responsible for dual-use research of concern oversight, and none have legislation requiring oversight of research involving especially dangerous pathogens or toxins. And I should mention all the kind of justifications for all these assessments are available on the website. So I encourage you to walk through them, see why we scored it the way we did. Countries, really do need responsible agencies and a clearer definition of dual-use research of concern oversight, and this legislation to back it up. Continental bodies like Africa CDC are helping provide examples of what this legislation can look like, including through the development of their regional biosecurity and biosafety legal framework. And then finally, probably the one that's kind of most relevant to the conversation today is our recommendation to establish a continental AI safety institute with responsibility for biosecurity and biosafety governance of AI-related tools and technology. As we have talked about, AI can help close the capacity gaps that we're tracking in the index, especially, for example, in medical countermeasure development. and biosurveillance. We're already seeing a positive trajectory in local MCM development across the continent. And AI-enabled drug discovery can just accelerate that progress further. So it's really important that AI be, adopted and used. Also for biosurveillance. AI-enabled outbreak detection can provide earlier signals, faster analysis, and better use of limited health data and personnel if incorporated effectively into systems. But the same tools that create these opportunities introduce new risks. So governance really needs to be part of that conversation from the start and not kind of like added on afterwards. So that's our case for a continental AI safety institute, because alongside these risks should be governance strong enough to manage them. And policies that promote responsible use, just like I talked about in the DNA synthesis screening portion, don't just make Africa safer, they shift the global marketplace toward tools that are safer everywhere, not just regionally. So you can have quite a bit of influence on how these tools are developed, rolled out globally. And these norms for responsible use of AI bio capabilities are taking shape globally. And so having an AI Safety Institute allows African voices to be at that table and not have to adopt standards or practices that are developed elsewhere. And the concern about misuse of AI bio capabilities and AI generally is not just a Western concern. A University of Cambridge study just found, for example, that in Northeast Nigeria, Boko Haram is already using AI to ideate potential attacks. And the concern, of course, is that interest will extend to AI bio capabilities. So having in place managed access and information sharing policies informed by a continental AC are really how we get ahead of and manage those risks. So my final slide. Maybe I don't have a final slide, but so thank you so much for this opportunity. And, the governance gap that I really highlighted at the beginning isn't inevitable. It is, a choice and one we can still get right. We have time, but we need to really kind of move now. And so we're continuing our work with the forum for developing and sharing best practices for effective AI bio safeguards, really forming the coalitions to move toward real governance solutions. And on, the Africa side, NTI Bio is working directly with African governments, regional bodies, and partners to help them, adopt the practices that we've put forward in the index and moving, from analysis to implementation. So we warmly welcome your help in closing these gaps, and thank you for the opportunity today.
Thank you very much, Ms. Severance, for that beautiful presentation. Please let's appreciate our speaker, Ms. Severance, for that. Let's show her some love. Use the emojis to say thank you. Thank you very much for that insightful presentation. Your presentation highlighted how rapidly and evolving these technologies are going, particularly at the intersection of artificial intelligence and life science, how they are creating new biosecurity considerations that require global action. Thank you once again. You also drew attention to the practical efforts NTI is doing, and we are glad that GET is also a part of this effort to strengthen biosecurity, including the development of standards of biological designs and all of that. Thank you very much. You also brought the discussion back to the African context that you talked about, the findings from the 2026 Africa Health Security Index, which shows that while biosafety and biosecurity capacities are improving across the continent, they still remain critically low. And you also highlighted the need for us to strengthen this laboratory safety, biosecurity training, regulatory capacity and the likes. Also, the recommendations you highlighted were very, very useful. You mentioned that we require DNA synthesis screening at the point of procurement. You also mentioned development of use research of concern, oversight frameworks to be implemented at the national level with guidance at the continent and regional level. Lastly, you mentioned that we should establish a continental AI Safety Institute with responsibility for biosecurity and biosafety governance of AI-related tools and technology. That was a beautiful presentation. Thank you once again. And as biotechnology continues to develop across borders, effective governance will require cooperation between government, scientists, industry, and international organizations and other stakeholders. So this brings us naturally to our next speaker, whose perspective will take us more directly into the role of biotechnology industry and the practical mechanism being developed to manage emerging risk. It's my pleasure now to introduce James Digant, who is the Vice President Policy and Biosecurity at Twist Bioscience, where he leads the company's engagement on biosecurity, regulatory policy, and responsible innovation in DNA synthesis. In parallel, he serves as chair of International Gene Synthesis Consortium, IGSC, leading industry body advancing harmonized DNA sequence and customer screening practice among gene synthesis providers worldwide. Through his dual roles, James has operates at the intersection of industry implementation, international standards development, and public sector engagement. He has been a central figure in advancing practical interoperable screening frameworks, working with government, standard bodies, and the scientific community to reduce misuse risk while enabling the continued growth of the bioeconomy. So his experience gives him a unique operational perspective on how global alignment of screening standards can close gaps, reduce fragmentation, and strengthen trust across borders. For today's discussion, he will be speaking on biosecurity in gene synthesis, strengthening screening standards, and responsible biotechnology practice. He will also be bringing perspective on emerging challenges associated with advances such as AI-enabled protein engineering and the implications this development may have for defensive biosecurity. Thank you, James, for accepting our invitation. We are glad to have you join us. The floor is yours.
Thank you very much, Ms. Adebayo, for that lovely introduction. I really appreciate it. Oops, I have shared the wrong window. There we go. All right. Well, thank you all for the opportunity to speak to you today. I want to give kind of some background on the biosecurity practices in the gene synthesis industry and sort of where all of that is going in terms both of first international standardization and then second kind of growth into defense against kind of emerging AI model capabilities. If you're not familiar with Twist, just a bit of background on the company. So we are a synthetic DNA manufacturer. We're based on the West Coast of the United States. We have facilities just outside of San Francisco and then just outside of Portland. The scale at which we manufacture, you can think of it as in 2025, we made almost 1 million genes during that year. And for this year, we're on course to make more than 1 million genes. So this is operating at a very large scale. And for each of those sequences that come in the door, we have to make decisions about, does that sequence pose risk? Is that customer trustworthy with the material that they've requested? And the goal really is just to make sure that all of that DNA is used responsibly. And Twist's sort of unique technological spin really comes down to a silicon technology that we have. You can think of it as sort of a very fancy inkjet printer where we're growing DNA up off the surface of a silicon plate. And so in the same physical space that you might be able to make 96 oligonucleotides, we can make more than 1 million. And so that lets us make about 10,000 more genes per unit of space, unit of reagents than other technologies, earlier technologies. And so we've used that technology kind of to drive down the cost of DNA per base, which has, I think, allowed scientists to run more experiments. But today, I want to focus on the fact that synthetic nucleic acids, DNA, RNA, these are dual-use materials. So these are materials that can be used for amazing good purposes, but also have some risk of being used for harmful purposes. So, for example, rescuing viruses and using those in inappropriate ways. And so we want to make sure that these technologies are used responsibly, and especially with the growth of AI biodesign tools. We've heard recently this phrase that you can think of DNA as kind of bridging the digital to physical divide. So you can go and use all of these AI biodesign tools and design some really impressive construct, but at some point you have to render that in the real world as DNA so that you can test your ideas and hypotheses. So that's kind of the role that the DNA synthesis industry is playing right now. Just some background on the current practices, so kind of we all understand what industry does today, so we can kind of contextualize that with where we think things should be going. So when you order something from Twist or really from many of the other major synthesis companies, there are two kinds of practices that go on. The first is customer screening. So this is really asking for verification of the fact that the ordering person is a legitimate scientist, they're at an institution, they have the facilities necessary to carry out the experiments that they have planned. We also do screen against some lists maintained by governments of sanctioned parties. We also limit the addresses to which we can ship. So we don't generally ship to PO boxes at all. And then only in narrow circumstances will we ship to private residences. Usually we're shipping to institutional addresses. So that's customer screening. And then the second part of our process is sequence screening. So it's looking at the sequence that has come in the door that we've been asked to make. And the goal for us is to understand, like, what is that thing? What is its biological purpose? What could it carry out if you put it into a cell? And then really, does that sequence pose any biosafety or biosecurity risk? We do this usually by looking at what the US government has called best match. So we take that sequence, we align it to a very large reference database of sequences, and we look for any window in that sequence, what is the best match in that giant database. And so if that best match is to some species that is regulated, then we treat it as sort of a sequence of concern. And then we ask questions like, is it legal for us to manufacture and ship that from the United States? Does it require an export license for us to ship outside the United States? For genes that are from controlled bacterial and fungal pathogens, we have an extra set of questions around, like, does that gene participate in the pathogenicity process? So for viruses, like the whole genome is dedicated to pathogenicity. So we don't have to worry about that question. But for bacterial and fungal pathogens, most of the genome really just keeps the organism alive. Only a narrow set of genes participate in pathogenicity. And so figuring out which those genes are, I think, is an ongoing challenge, which I'll talk about in a few minutes. So I wanted to give kind of a brief history of the way the global industry has kind of wrestled with these technologies and the implications that those technologies have for policy, and how all of that is kind of moving towards international standardization as the end goal. So if we go all the way back to 2002, these technologies were just beginning to mature, that you could start making longer oligonucleotides, short segments of DNA. And so an academic named Eckhard Wimmer used that to rebuild the poliovirus from scratch. So the poliovirus is a fairly small genome. It's 7,500 base pairs, but it took his lab 14 months to do this. And he purchased all of those oligonucleotides commercially, and then in his lab put them together to make this viral genome. And the political point I think he was trying to make, he was doing this during the World Health Organization's ongoing campaign to eradicate polio. And so he was trying to say, like, while you may eradicate the naturally occurring instances of this virus, technologies are now allowing us to kind of recreate that virus from scratch, even if it no longer exists in the world. And then in 2006, a reporter from the Guardian newspaper in the United Kingdom went to one of these kind of emerging gene length synthesis companies and bought what I understand to be one oligonucleotide that was a best match to the variola virus. That's the virus that causes smallpox. And then wrote this very nice article laying out why are these companies willing to send an oligo from smallpox to someone's flat, like his home in London. Great question. This had a very high impact, I think, on the policy community in many countries around the world, starting to think about the questions that these technologies might create in terms of how you ensure that they're used safely. So from 2007 to 2009, there were a lot of discussions among the companies that were capable of using these technologies to make what you can think of as gene length DNA, so around maybe 1,000 base pairs. This was just becoming possible at that point. And so the 5 biggest companies at the time got together and formed the International Gene Synthesis Consortium, the IGSC. The goal there was to understand that you're going to have companies around the world that are very fierce competitors in the marketplace, but that we all have an interest in making sure that our technologies are not misused, as do the governments and citizens of the world into which we sell these products. And so, given that degree of agreement, we all want to get together and ensure that the practices that we have for screening our orders are sort of state of the art. And then from like 2010 to 2023, there was a time in which the US government issued guidance documents. That guidance at a very high level suggested, but did not require, that companies that have these synthesis technologies do 4 things. 1, screen your customers, make sure that they're legitimate, and make sure you understand who you're selling products to. 2, screen your sequences. And in 2010, they said you only need to worry about things that are over 200 base pairs at that time. But make sure that you can identify any sequences that might be, of concern. 3rd, if you do find something that's strange, alert law enforcement. Call people who can respond to that concern. And then keep all of your records for 8 years. And the reason for 8 years at the time, the what's called the statute of limitations in the US for use of a weapon of mass destruction was 8 years. And so the thinking there was that the company should maintain all of these records in case someone did misuse a biotechnology, and we had to go back forensically and figure out how did they get their hands on that. That same time period, 2010 through 2023, saw the technology really improve at a rapid clip. So Twist was founded in 2013. Lots of different companies and technologies. There have been a lot of focus on development of benchtop devices, so very small format devices that could sit on a benchtop in a laboratory and synthesize gene-length DNA. So in 2023, the US government issued an update to their guidance. And they said 2, among other things in the document, they said 2 what I think are critical new things. 1, that these benchtop devices should be held to the same standards as the sort of traditional centralized screening providers. That everyone, regardless of the physical format in which you make DNA, you should ensure that it's not going to be misused. And then the second thing that update said was you actually need to care about any piece of DNA down to 50 base pairs. And the reason for that being that the technologies for assembling oligonucleotides into longer and longer genes had gotten much easier. And so more attention should be paid to orders for smaller pieces of DNA, that people could sort of hide their intent to acquire something that maybe we wouldn't give them if they just ordered 1,000 base pairs, but they might hide that intent by ordering smaller pieces and then themselves stitching that together. 2024, you'll see the theme here is that things have really started to speed up. So in 2024, the US government released a framework. The United Kingdom released a guidance document that adhered quite closely to some of the ideas in the US framework. And so you're starting to see sort of like international agreement on some of these things are as best practices. The framework in the United States for the first time limited US dollars that the US government was spending for scientific studies had to be, if you're going to buy synthetic DNA, you had to buy it from a company that publicly stated that they did these practices that the government recommended. And so you'll see screen sequences, screening customers, reporting suspicious orders to law enforcement, retaining records. They did add an additional one, which is ensuring cyber and information security. So our customers trust us with a lot of like their very important intellectual property in the form of these sequences. And so there's a lot of responsibility on our shoulders to make sure that we protect all that information and have appropriate cybersecurity and information security practices in place. And so this was the first time that a government had said, you know, we're willing to try and incentivize companies to put these practices in place. We're still not going to require them, but we're going to try and incentivize them. In 2025, new administration came into power in the United States. They have been rewriting the 2024 framework. We understand the goal is still to limit federal funds to companies that have these biosecurity screening practices in place. One of the new goals might be to introduce third-party verification. So as I'll get into, a lot of what the industry has been working on the past 3 or 4 years has been trying to standardize the way that we measure whether these screening systems are doing the right thing or not. You know, how accurate are they? And once you have a standard, then you can measure against that standard. And so I think we're at a point where we can now start to engage in third-party verification of our screening systems. And then in the EU, you're starting to see drafts of what they're calling the EU Biotech Act. And there's in the recent draft, there's a chapter there around possible requirements for biosecurity screening. So I think governments are really starting to wrestle with, should we move from a model in which we kind of encourage this to a model in which we require it? And some of that has been driven by the growth in AI design tools, opening a lot of new questions around sequence screening and what kinds of threats we should be looking at. Some of the work I think that industry has been doing in parallel has really focused on trying to develop standards. If we went back maybe 4 or 5 years ago, one of the things we wrestled with was that every company would kind of implement their own strategy for screening, and there wasn't really a way to measure yourself against an independent benchmark. So you'd invest all this money in a screening system, and then you had no way of assuring yourself that you'd actually done a good job. And so we worked with the National Institute of Standards and Technology, NIST, in the United States, to develop an approach to standardizing the creation of test sets and then a way of measuring against those test sets. And so NIST now has a public example available of a test set. So anyone who's building a screening system can evaluate that against a public example. And then there's a process that's being put in place, work between the IGSC, between the National Institutes of Standards and Technology, and with some third-party organizations to have monthly cadence creation of new test sets, and then the ability to evaluate your screening tools against those new test sets. And so the IGSC is now trying to drive our membership to at least once a year go ahead and evaluate one of these test sets and have a third party say, yes, you know, you have passed the level at which we think screening needs to be implemented to keep an order stream safe. And then that work has continued and resulted in some publications with NIST around how do some of the screening tools look at, you know, if you have a protein that has been heavily engineered by an AI design tool, and then you determine whether a screening system can detect that or not, how then can we check to make sure that that heavily engineered protein still is functional? And so NIST has been doing some really important work in that area, designing laboratory protocols to evaluate the functionality of some of these heavily engineered sequences, and then map that back to how we should think about standardization of detection. As part of that work in generating test sets, a long-standing issue that came up both from the industry side and from governments has been, and I sort of hinted at this earlier, which is from these bacterial and fungal pathogens, how do we get an understanding of which of the genes are involved in pathogenicity? There is no magic list. Some governments maintain lists of species. So they might say, for example, Bacillus anthracis is a control organism. But Bacillus anthracis has a very large genome. There's only a few, maybe a dozen or so genes that actually participate in pathogenicity. Who is responsible for going in and calling out that gene level information? And the answer globally has been nobody for a long time. And it's something the industry has consistently asked governments, like, we would like governments to have an opinion on kind of this, what we refer to as component level biological risk. What are the genes that are really participating in pathogenicity? And so as part of that test set creation process with NIST, the IGSC and an organization called IBIS, the International Biosecurity and Biosafety Initiative in Science, spun out a new consortium. So this is the Sequence Biosecurity Risk Consortium, SBRC. The goal here is actually to be much broader than the gene synthesis industry. So it's to bring together experts, virologists, bacteriologists, mycologists, who are all willing to kind of come up with a collaborative joint annotation for which of these genes are bad genes, which of the genes should be, and you can see here in the graphic, considered something that should be flagged by a process that is looking for biosecurity risk, or something that is known to be safe and can be categorized as a no flag, or that we just don't have enough published literature on and should remain in some kind of middle zone of being undefined. And so it's really trying to parcel out like human knowledge about component level risk. And I think this is an incredibly important ongoing effort. The goal of the consortium is to be as broad as possible. So if you are interested in lending your expertise to that consortium, I'd encourage you to go visit sbrc.bio. I think right now the gene synthesis industry is really benefiting from the kinds of expertise that have been shared in that forum. And the output of that consortium is a growing, what they refer to as a biosecurity flag rubric. So a set of categorizations on this component should trigger a flag, this shouldn't, this is undefined. And that's ongoing work. And then all of this, I think, is heading in a direction where now that we have, an initial standard coming out of at least one standards body, now that we have a growing consortium that can point to component level risk definitions that are agreed upon by many experts, now that we have companies subjecting themselves to kind of third-party assessment against these standards, now I think we're gaining a lot of confidence and moving towards making this an international standard. So taking this, for example, to ISO and trying to create a formal international standard around current best practices in biosecurity screening. And so IBIS has been playing a leadership role here. The IGSC companies are extremely supportive of this effort, and we really look forward to IBIS putting in place this ISO standard, working in the ISO process, which requires standard setting bodies from across the world to sort of weigh in and have a voice in that process. And so hopefully within a year or two, which ISO is not a very fast process, but I think it's a broad convening process. We'll have an international standard that will sort of encode these best practices. And the IGSC is already quite an international organization. So we have right now around 50 members from 10 different countries. And so the IGSC is also looking to actively expand in support of promulgating adherence to these kinds of international standards. And then the last thing I think I want to say is that while we're working to encode and standardize our current practices of sequence similarity-based screening, we also recognize that AI models are kind of making all of this much more complicated at the same time. And so we're in parallel engaging with a lot of technology domain expertise on the model side, on the AI side, to develop defensive AI model-driven ways of protecting our order streams from highly engineered or entirely novel protein designs. So we recently published last year some work with Microsoft and the IGSC on what they called the Paraphrase project, which was using AI models to highly engineer controlled toxins and viral proteins. All this work was in silico. It wasn't done in the wet lab. But then seeing which screening tools could detect those highly engineered proteins and at what degree of engineering would the ability to detect those things break down. And so that work was published in Science, and Microsoft has some very fun visuals that they put together to kind of describe that research. And we're looking to certainly continue these kinds of ongoing collaborations to put these defensive models in place and to have them keep pace with the advances in the AI protein design models that continue to get better and better. So with that, I will thank you all for the opportunity to present to you today, and I look forward to both the remaining speakers and the Q&A session.
Thank you very much, James, for that beautiful presentation. Please let's show James some love. So thank you to him for that amazing presentation. That was quite an insightful one. So we were grateful for that. So you particularly mentioned and took us through the evolution of biosecurity in the gene synthesis industry. And that was quite a good one, from the early concerns such as 2022 synthesis of poliovirus through the establishment of the International Gene Synthesis Consortium in 2009, and to the more recent and international screening standards. Thank you very much for that. You also gave us a valuable look at how screening works in practice, including customers verification, sequence screening, the regulatory checks, and you made it clear that we must continue to evolve because AI are now capable of redesigning existing proteins and generating novel biological sequences, which means that traditional screening approaches will face new challenges. So we need to become more ready for them. Thank you very much for connecting the history, the current practices, and the future challenges of biosecurity in gene synthesis so clearly. Thank you so much. We are grateful. Right now, so we've discussed the safeguards surrounding the synthesis and the access to biological materials. We will now move to another critical part of the conversation, which is the research itself and the responsibility of scientists and institutions to recognize and manage potential dual-use risk. So our next speaker brings decades of experience in virology, biological research, international scientific adversary process, and disease eradication. Mr. David Uleto is a senior principal scientist at the Defense Science and Technology Laboratory, where he researches large GNA pox viruses and small RNA alpha viruses. His research interests include virus morphogenesis and cellular metabolism, with a strong focus on the eradication of serious viral diseases and preventing their re-emergence. Since 2020, Dr. Uleto has served as an advisor to the World Health Organization on veillonella virus research, and since 2009, he has advised the FAO and OIE on ridden pest virus research and control. He is also a founding member of FOA/OIE Rinderpest Joint Advisory Committee, and in 2017, he was appointed to the UK Scientific Advisory Committee on Genetic Modification. His extensive experience places him at an important intersection of scientific research, biological risk, international adversary processes, and responsible governance. Today, Dr. Ulet will be speaking to us on governing dual-use research, building a culture of responsibility in life science. His presentation will take us beyond the question of what safeguards are needed and into the equally important question of how we build a culture of responsibility within the scientific community itself. Thank you, Dr. Oleto, for joining us. Thank you for accepting our invitation. We are so glad to have you. The floor is yours. You are muted. We can't hear you. No, we cannot hear you. Do you mind reconnecting your mic again? Hi, Dr. David Duleto, we cannot hear you. All right, that's fine. You can reconnect and disconnect and reconnect. Thank you. Okay, for us to go to the next speaker. All right, that's perfectly fine. Thank you very much. We would come back to your presentation immediately after we have our next speaker present. Thank you. Okay, moving on to our next speaker. Our next speaker for the day is Dr. Ketan Trot. He brings a distinctive perspective at the intersection of science, policy, regulation, biosecurity, bioethics, and emerging biotechnology. Dr. Ketan Trot is a scientist and policy professional working across scientific research, regulatory compliance, institutional governance, international cooperation, and science communication. At BRIC in STEM, he contributes to regulatory compliance, ethics, and governance around advanced biomedical research. He has served on high containment laboratory validation and assessment committees of India's Department of Biotechnology and contributed also to ICMRI's National Ethical Guidance. During his tenure with the Department of Biotechnology, he managed international research and innovation partnership spanning 18 countries and more than 100 bilateral projects. His work also extends into emerging frontier of biotechnology. He is a track lead for policy, regulation, and standards under India's national strategy for orbital biomanufacturing and the space bioeconomy, and has also worked on issues including space biology, planetary contamination, bioethics, biosecurity, dual-use biotechnology, DNA synthesis, screening, and anticipatory governance. Dr. Trott will speak on emerging biotechnology and responsible governance, anticipating risk and enabling sustainable innovation. Dr. Trott is also an alumni of the Youth for Biosecurity, and we are so glad to have him here. Thank you so much, Dr. Trott, for honoring our invitation. The floor is yours.
Thank you so much. Faith, are you able to see my screen?
Yes, you can see it.
Okay, great. Hi, I'm Ketan. I am a scientist and a regulatory compliance officer at a Government of India institution here in Bangalore. And I'll be talking about emerging emerging biotechnology and a responsible governance. So this word responsible is something I have been thinking about a lot in my career. As I started my career in the regulatory governance of biotechnology, I increasingly realized that a lot of the discussion is happening in pieces. The entire field of research and innovation in biotechnology is not being uh, uh, governed from a lens of a collective responsibility, instead of as every time there is an emergence of a new a biotechnology, or if any other technology is converging with a biotechnology, we uh, uh, we are responsive in our approach, and we start addressing only that aspect of bioresponsibility. However, so in today's talk, I'll try to put forth an argument that we cannot separate a biosecurity from other aspects of a bioresponsibility, which include biosafety, bioethics, and the society as well. And in the later part of the talk, I'll also mention a couple of emerging convergence of technology, which has a lot of implication on our sustainable as well as equitable future. So I will also try to convince that there are frontiers of biotechnology which are evolving in a direction in which, in my opinion, Africa needs to engage early in order to ensure that they have an equitable contribution, and uh, your voices are also heard early in shaping how the regulatory governance of all these spaces would look like. So, as you know, that our biotechnology is becoming more powerful, accessible, and interconnected. In the today's talk itself, in the webinar, we heard about how the synthetic biology and AI bio the critical challenges in these two are being addressed, and I'm sure we'll also heard about other aspects of these uh, convergences. However, there are some emerging bioresponsibility challenges also in automation. Also, as we start looking at space as a location for a bio manufacturing, who will have access to which orbits, and how much of the lower Earth orbit can be exploited to do the space bio manufacturing, as the ability to access the space is still limited to only a handful of countries across the world. Then who gets to decide how much of the space belongs to who? So also I would like to talk about on that in this a larger discussion around a bioresponsibility, the important aspect of the climate change and its impact on ecology, and how it will change the ecological dynamics of the vectors and our carriers, is also something which is not being addressed to the extent it should be. So I would in this talk, I would also try to invite the attention on all these topics, and also highlight a couple of the initiatives which we have started here in order to address some of these governance challenges. So as Haley also mentioned, the biology is evolving extremely fast, and it is also converging with a lot of emerging fields. Therefore, the how we approach the governance of this emerging biotechnology, it should also converge and look at it collectively. So, how traditionally we have responded to the emergence of the new biotechnology has been that, like, after a discovery has happened, the regulators step in, and we go back and forth and engage the policy makers as well, in order to create policies which effectively then impact how a particular technology will play out in the world, and be experienced by the society. However, in my opinion, I think the model we require is that a concurrent a discussion across the scientists and regulators and industry, and the security agencies, and the public health bodies, ethicists, the communities themselves, and how also the experts on the environment, to understand how exactly all these emerging technologies will engage with our societies and the environment as well. I'm highlighting the importance of engaging with the communities and societies also, because each society evolves very differently, and their priorities and their approach to all these challenges, and how all these technologies are going to affect them. All their understanding of these technologies could also be very different, and it's important to have a consolidated understanding of how the different communities are also looking at it, in order to define a regulatory approach to a particular emerging technology. So, and why is this important is also because the emerging risk often involve a lot of uncertainty. It's not always simply the unknown hazards, and as a policy advocates, it's not always possible for us to anticipate how exactly will a policy or the technology itself will play out in the real world scenario. It is important to acknowledge that different stakeholders see a different risk, benefits, and values. So it's important to have them at the table, and also incorporate all of their understanding also into defining how the regulatory mechanisms would look like. Also, it's important to acknowledge that more often than we would like to believe, those affected by an innovation may not be the people who are designing that innovation, or the or the regulatory policy. Uh, uh, Engagement with these broader stakeholders can help identify our concerns that only a technical assessment, a risk assessment alone may miss. And also, we need to anticipate what would be the biosafety, biosecurity, bioethics, and also the societal challenges which will be opposed by a particular technology as it is developing and evolving. We need to anticipate, and the governance should begin even before deployment, and not after a particular innovation has happened. And it's not just me, but like including the WHO also describes that the biorisk management and the DURC is a shared responsibility, and it should involve our policymakers and our regulators and our scientists, institutions, our funders, our publishers, our security actors, and the our private sector actors, along with other stakeholders. So in this other stakeholders, I would say that it is also important to have the participation of the society which will be impacted by these emerging technologies. So I'll just offer you a particular example. A lot of investment was done in R&D of generating the mosquito gene drives. So now gene drives are these genetic elements which are not only heritable, but they also completely replace a particular gene with a gene of interest in the entire population. So they have the a possibility of not only introducing a new gene in the population, but to completely replace an existing gene with a synthetic gene in the entire population in the wild. So when this technology was being developed, of course, the intention itself was right. It is important for us to control mosquitoes as a vector. However, after investing a lot on this technology, we started acknowledging the bioresponsibility challenges it poses, including biosafety, which includes what exactly would be the ecological effects of a gene drive. Is there a possibility of any non-target effects? Is there a gene flow? The entire the fact that any introduction of gene drive into the wild is irreversible on the bioethics, and it was important to understand who will decide if those if these mosquitoes can be introduced in their environments, what level of uncertainty of how it will play out in the real world scenario is acceptable, what exactly constitutes an informed consent of the entire society. So is the society okay, or is the society open to having an innovation go out in the wild, which is completely irreversible? Uh, of course, it involved the biosecurity risk as well, because of the dual use nature of the technology, and like who will govern the research and the material was also another important aspect. However, the it is also important that from a societal perspective, to ask that who will be the beneficiary of this intervention, who will bear the potential risk if anything goes wrong. And it is also important to ask if the potentially affected our communities, how will they be consulted and meaningfully involved in the discussion, and how this our technology will play out in the wild. Because of this, even after extensive R&D on a gene drives in mosquitoes, there are absolutely no examples across the globe where this technology has been actually used in the wild. So also, as I briefly touched upon that, after COVID, at least in my opinion, I've seen a less and less a discussion about climate change. However, the climate change is still happening. As some of you may know that, like the last year itself, the global average our temperature, it was already 1.44 degree Celsius, more than the pre-industrial levels. Also, the last 11 years have been the hottest on record. My our concerns about how this climate change is engaging with a bioresponsibility or a bio a security is to ask that across the globe we are building a massive amount of infrastructure in terms of the BSL 3 and BSL 4 labs. As the climate is changing, it means that we will experience across the globe a more intensity and a more frequency of extreme weather events, which includes the heat extremes, heavier precipitation, which could lead to flooding, stronger tropical cyclones and hurricanes and wildfires and the compound flooding, etc. How it will impact all these laboratories, and it's also important to understand. So a student of mine did a study on this, that if uh, as per the IPCC predictions for any increase in the heavy precipitation across the globe, how many of high containment labs across the globe are at will be at a higher risk of experiencing a flood situation because of heavy precipitation in that area? It is important to anticipate that if there is extensive, unprecedented flooding within a high containment lab, it poses a massive risk of uh, release of any of the these high containment microbes which are being handled in that laboratory. And also, when the a natural calamity has already hit, the entire response infrastructure of that locality is already extremely strained. In that case, if a high containment a laboratory also ends up compromising the containment because of flooding, it's important to have a very clear a protocol and on how exactly such a risk will be mitigated, right? Similarly, as I also spoke about the a possibility of a severe windstorm will also increase across the globe. So the highlighted areas are the regions in which there is a medium confidence of increase that it will have a much more severe windstorms in the future. It's also important for us to ask, are our laboratories built to withstand not only the current climatic risk, however, they should also be prepared in terms of any increased intensity of climate related risk in the future as well. So apart from this, I would also like to highlight that because AMR is also considered as a massive concern globally, at least in terms of health security. It's not just we are not the only victims of AMR pathogens, but because of our anthropogenic influence, we have also contaminated one of the most remote places on Earth irreversibly to uh, uh, AMR microbes. As you can see the example that we have already introduced drug resistant enteric bacteria into Antarctic ecosystems, and they are already affecting the seabirds and seals as well. It's not the only location. Even the subglacial a lake Vostok is also already contaminated as around 50 contaminant phylo types have been already identified in the drill bit and borehole when our samples are taken of the frozen lake. So apart from that, it's not that we have only contaminated the most remote places on Earth, but we have also contaminated the moon. So there was an Israeli lunar lander mission which was launched in 2020. This lander was supposed to have a soft landing on moon. However, accidentally it crashed onto the moon, and it was one of the first time a private industry was sending a lunar a lander mission, and one of the experiment on the mission was to understand how exactly these are tardy grades, which are these microscopic organisms, which are polyextremophiles. It means that they have the ability to survive extreme desiccation, freezing temperatures, and high doses of ionizing radiation. We accidentally, we as the species, I mean, we accidentally ended up introducing or contaminating the lunar surface with these tardigrades as well. And it is still unknown how many of them have survived, and how exactly are they right now after the crash on the moon surface. Uh, I would just like to highlight uh, on this that with the increasing discussion on the orbital bio manufacturing, like the countries which have these space-faring abilities are having a very uh, strong hold on how the policy would look like, because under the outer space treaty, there is no international organization which governs exactly how the space security is being upheld, and how the coordination across the nations of the globe would look like. Increasingly, we are seeing again a formation of two blocks across the US and the China, which are trying to independently define how the regulation of accessing these space and all these celestial bodies would look like. One important example is, of course, the Artemis Accords, in which 7 out of 70 signatories are African countries. However, only a handful of countries were initially involved in actually drafting the document of Artemis Accords. Everyone else had just only become a signatory to it. So in my opinion, it was not really a highly consultative process. So I would invite all of you to also think about engaging early in this field, because as the access to this technology increases in the future, it's important that the equity to the uh, uh, like the larger globe is also ensured, and it doesn't happen that the countries which have more access to the space occupy majority of the resources, leaving the countries who do not yet have such capabilities with a very few or uh, undervalued resources in the future. So as and how are we addressing this? As I started working on this, I realized that the understanding of bio responsibility is not only missing in the policymakers. It is also a lot of researchers, a lot of PIs in the R&D institutions across the globe also lack the understanding of why exactly responsibility is important in life sciences research. So in order to address that particular challenge, uh, we have created this a bioresponsibility collective, which comes up with a comic every month to explain emerging issues in um, uh, the uh, emerging issues in biotechnology, including the AI bio and mirror our life. And our upcoming our comic, of course, is on the planetary biosecurity. So I would invite you to take a look at this. And the idea of all these comics is that if I have to explain a complex biosecurity idea to a policymaker who may not always have a technical background, uh, in that case, I can use these comic as a tool, as a visual a tool to explain it to them more effectively. With this, I would like to offer a few updates that a colleague of mine and I have created, like a resource portal to understand all these aspects of bioresponsibility, including academic integrity and research ethics as well, which
Hey, Dr.
I believe is also extremely
Do
important.
you hear me?
Yes, Faith, are you trying to say something?
Okay. Do you please round up your presentation so we could move to other speakers?
It is the last slide. Just
Oh, okay. Beautiful. Thank you.
I'm done. So there is we are organizing a conference in February next year in India. It will be the first of its kind conference in India, and we are expecting a strong participation from the global south. So I invite all of you to please check it out and do apply. And we look forward to a meeting at least some of you in February next year. Thank you.
Thank you so much, Dr. Church, for that beautiful presentation. Please let's show some love to him. We are grateful. Thank you for honoring our invitation. Without wasting much more of our time, we still have two more speakers to present. Dr. David, are you ready for us now? Please confirm if you can hear me. Dr. David Uleto. Thank you very much. Dr. David, are you there? Okay. Yes, thank you. Please confirm if you can speak now.
Hello, can you hear me?
Yes, we can hear you.
Oh, hooray.
Yes.
Do you want to put the slide deck up for me? Because I
Yes,
can't
1
drive
minute.
it myself. Yep, brilliant, thank you. And I'm very sorry for the earlier issue. I don't know what it was, but I'm glad to be able to speak now when we get into presenter mode. So thank you to Faith, and thank you also to Haley and James and Keaton for really very illuminating presentations which they gave. So my presentation is governing dual-use research, building a culture of responsibility in the life sciences. Next slide, please. And essentially, this is just for us to consider why responsibility matters. So life sciences have transformed all of our lives throughout history, in all sectors, whether it's health, public health, agriculture, industry, and life sciences will continue to transform our lives. Keaton alluded to climate change, so our environments are going to be changing, our economies are going to be developing, and our populations increasing. These changes and challenges will require life sciences advances to help us maintain and improve our lives and lifestyles. And the sophistication and quantity of life science research is also increasing. Next slide, please. So some of the earliest life science outputs in human, in effect, prehistory, were the use of plant toxins for medicine, arrowheads on arrowheads for hunting. And this is clearly a dual-use technology. And what I'm driving at here is that dual-use technology is not a small subset of technology. It's actually a very big subset of technology. And the scale and complexity of biological processes in modern society increases the scope for the misuse of life science advances. So essentially, our problem space is that scientific progress brings enormous benefits, but people can misuse those outputs to cause harm. And this brings challenges for society, and it brings responsibilities for members of society. Next slide, please. And I just want to bring up, just gently touch on the difference between dual use. If we could go to the next slide, please, Faith. The difference between dual use research and dual use research of concern. And just in a nutshell, dual use research of concern is a small subset of research that has a clear potential for misuse. An example of this would be research that overlaps heavily with high consequence pathogens, for example. And I think James alluded to that when he talked about the synthesis of poliovirus, a template-free synthesis of poliovirus, and of course we had a template-free synthesis, more complex synthesis of a pox virus a few years ago. But dual-use research, that is not research of concern, is research that could potentially be misused for harm, and there isn't necessarily a clear path to misuse. Next slide, please. And so dual-use research of concern is often big enough to see, which gives us an opportunity to regulate it. But dual-use research is less obvious, it's more amorphous, and governance is thus more difficult, more challenging. And so because of that, the responsibility for safety extends well beyond the regulators. And this graphic I have in the bottom right corner there is just a graph drawn from some data I pulled from PubMed. So this is a database effectively of all medical research and therefore life science research globally. And you can see that from the 1950s, when there were tens of thousands of publications per year, we are now in a situation we are in the multiple millions of publications per year. And you see, apart from a little blip that comes immediately after the COVID pandemic, we have a very steep line of increase there. So the volume of research and the number of researchers producing it is increasing, and that increased research means increased dual use research, which is going to stretch our capability for governance. Next slide, please. And so we have challenges for governance, and these challenges themselves are also increasing. The previous speakers have mentioned the convergence of AI and biotechnology, and AI-enabled design lowers the bar for biotechnology and synthetic biology experimentation. Now, this could and it should be an enabler for development in lower and middle income countries. It could and almost certainly will accelerate the number of the increase in the number of scientists who are active in biotechnology, and further, and it will accelerate the increase in the quantity of outputs. And in addition to AI, we have laboratory automation, which synergizes with AI to increase the volume and reduce the cost of high throughput research in biotech, which again will accelerate the increase in the quantity of outputs. And one of the things that we don't often think about is that AI developers often come from a computational science background, and they have a very different safety and ethical culture to life scientists. They don't have the ingrained culture of risk assessment that life scientists have had since the 1970s with the advent of molecular biology. But they are increasingly active in the life science arena, and this creates an instability in governance because there are people who aren't necessarily aware that there is governance, which leads us to ask, are our governance approaches future-proof? And we heard, certainly from James, that governance is not standing still. But as I think all three of the previous speakers said, there are issues as to whether governance can keep up with the advances in research. Next slide, please. And so governance has specific challenges. The rapid pace of technological advance, the uncertainty over the nature of future risks, which Keaton certainly touched on, international differences in regulations, which I think Haley and James both touched on, and balancing security with openness. And there are ethical challenges, freedom of inquiry. A lot of our most profoundly beneficial advances came from scientists who were just doing stuff that they thought was important, in some cases thought was cool. And as we go forward with new governance, we have to be very careful that we don't over-constrain the ability of some of our brightest and best to actually produce the benefits, the future benefits that we are going to need. And we have the challenge of balancing that with public safety and also global equity, and making sure that we don't leave poorer countries behind. And that leads me to the conclusion that traditional regulation is not enough. If we go to the next slide, please. What I'm driving at here, next slide, is that rules matter, but culture matters just as much. And culture is about personal responsibility. And if we go to the next slide, please. This graphic was something that was drawn up, a committee in which I was involved for WOA, which used to be the OIE, and their guidelines for research. If you could just hit next slide, please, Faith, and that will bring that graphic up a bit bigger for people to see. And this is a graphic which talks about all the various people who are involved in risk assessment. And at the first part of that, at the research proposal, is the people who are designing the research, the people who we broadly think of as scientists. But there's also an institutional reviewing body. There are the funders. When it comes to the research, there are the junior researchers undertaking this. There are the collaborators who help in the analysis of results, and there are the people who are involved in publication. And all of these people at all of these stages have a responsibility in assessing the risks that come from this research. And there is another group who are involved, which is the public. So if we go to the next slide, please. We would say we have these responsibilities. So who is responsible? My take on that is everyone. But when do we learn that we have responsibility for safety in science? Where do we teach that? And my view is we should be teaching the responsibility in science wherever we actually teach science. If we teach science in primary school, and we do, we should be teaching responsibility in primary school. This needs to be age appropriate and it needs to be context appropriate, but we should be teaching it in primary school and high school and onwards. And so we should be developing resources for teaching this, teaching responsibility in science at all levels. So when we arrive at the start of our career in science, as a young researcher starting out on a PhD or just joining a laboratory for the first time, we must already understand at that point that we have a societal responsibility for safety. And if we go to the next slide, please, Faith. I feel this is so important that I put this slide in two different ways. Responsibility is about ethics. Where do we learn research ethics? And I think too many people learn about research ethics when they have to write an ethics statement. And that's too late. Research ethics needs to be taught wherever science is taught. And I know I'm repeating myself, but it's so important. It should be taught in primary school and high school and onwards. And that way, ethics gets embedded before we get to professional status. And it also means that non-scientists, people who don't go into a career in science, but who may have some, maybe lay people on an ethics board, also have some understanding of research ethics from their primary and high school time. And that takes me on to my last slide, which is about the public. And I want to repeat a phrase that Ketan mentioned. You cannot build responsibility without building understanding. And our biggest stakeholders in the science enterprise are the public. They have the most to lose from accidental harms and deliberate misuse, and also the most to gain from the benefits. So they are the major stakeholders, but they are not part of the scientific enterprise. They depend, however, on scientists to inform them of the risks, and they depend on the other stakeholders, the fund holders, the funding bodies, the institutional review bodies, to protect them against harms. Research that is approved is undertaken in their name, and there is a duty of transparency towards them. But there are challenges in framing communication to be easily understandable. Effective public communication is vital to the success of the scientific enterprise, and a societal culture of responsibility where everyone, including non-scientists, has some knowledge of research ethics is vital for that effective public communication. And I see I've got a typo there. I say effective publication. I mean effective public communication. And this requires wider engagement beyond scientific community. And I'm not aware that any nation is actually consciously doing this, building ethics in a primary school. There may be schools who do this, but I don't think it's done. I don't think it's given in guidance from ministries of education. And so this is where I say this requires wider engagement beyond us. And I think it's relevant to all nations that we need to be taking that message out to our ministries of education and asking our ministries of science to take this message out to our ministries of education so that we can build that in. And that will help plug some of the gaps that legal frameworks and governance will be slow to plug because they don't move as fast as science itself. And I think the next slide is just some further reading. And I think that's the end of what I have to say. So thank you very much. I hope I've gained us some time back, Faith. Thank you.
Thank you very much, Dr. David. And we cannot address every emergency to the French speaker for today. We apologize sincerely for the time. We might not be able to finish at 2 p.m. West Africa time as promised, because we still have one more speaker, but we'll be as fast as possible. We have Dr. Mary Asthma. She's a public health manager and virologist with extensive experience in research, biosafety, disease surveillance, discussion. Dr. Mary, we are pleased to have you. You now have the floor. Dr. Mary, can you speak now? Please confirm if you can hear me.
Hello. Hello. Hello.
We can hear you. Please go ahead, Dr. Mary.
Okay. Thank you to giving me the floor. I will focus on how we can balance innovation and governance sustainability, strengthen biosafety. Okay. I will focus on how we can balance innovation and governance to sustainably strengthen biosafety and biosecurity in African laboratories. To illustrate this, we will follow one sample journey from the bench to policy. Let me start with a story of one journey samples. At hour 0, a district hospital reported 9 cases of unexplained fever. Samples are collected. At hour 18, the cold chain is maintained, triple packaging is respected, and the sample arrived safely at the National Reference Laboratory. At hour 48, the genome is assembled, a new variant appears. And hour 672, data were safely shared through the approved platform. An alert is raised and the ministry activates a response. This single journey shows why we need both innovation and governance. We have some questions. And at hour 0, was the staff trained? Was PPE available and correctly worn? At hour 18, the cold chain and hour 18, this is where innovation and governance meet, because during innovation, we have better diagnostics and governance here is safe transport. At hour 48, we have some innovation. Innovation shows us what we are facing, but governance asks who has access to this sequence? How is securely stored? This single journey shows us that without innovation, we will still be blind at hour 48, and without governance, this sample will never reach the hour 80. This is here the balance we must strengthen in our African laboratories. Oh, I have, okay. We have to understand that African laboratory is going through 3 major waves, including manufacturing. Wave 1 is manufacturing. You are building capacity to produce vaccine and biologicals. Wave 2, we have genomic surveillance networks, and wave 3, we have frontiers. It's better to know that each wave brings incredible innovation, but each level also brings a new level of biorisk. And that is why our biosafety and biosecurity governance must evolve at the same speed as our innovation. Let's now look at the same sample journey, but this time, let's read it as a risk chain. We have 6-step sample collection, unprotected staff, transport, broken cold chain, containment, uncertified biosafety, sequencing, we have cross-contamination, data, we have cyber unencrypted genomes or cloud storage outside. 6, we have publication. We have a dual use method released without institutional review. We have a structural reason here. The risk is not concentrated in a high containment room. It is distributed across every hand over. Sorry for. To govern the risk, To govern the risk chain, we need to be clear on concept. And we have three concepts here: biosafety, biosecurity, and dual use. Biosafety is protecting people from agents. It concerns Pillar 1. Biosecurity is protecting agents from people, and dual use is governing knowledge itself. Biosafety prevents accidental release. Biosecurity prevents intentional misuse, and dual use prevents irresponsible dissemination. Why controls do not halt? We have 4 recurring gaps. Gaps 1, we have no legal mandate. Many member states still lack dedicated enforcement, biosafety, and biosecurity law. Gap 2, we have no workforce. The biosafety officer role is not too often an unpaid additional duty with no certification. Gap 3, we have no recruiting resources, and gap 4, no cross-border alignment. The good news is that we have the African CTC model legal framework. Let me give a real-world dilemma in this diapositive. A postdoctoral
We can no longer hear you, Dr. Mary. Please confirm if you can see you.
I have issues with the connection. Have you seen my screen?
Yes, we can, but it's not on presentation mode.
I did not see my own screen. Let me give a real-world dilemma in this diapositive. A postdoctoral fellow in your institute has optimized an amplification protocol that makes a hard consequence pathogen far easier to detect, and with the same published steps far easier to produce at scale. The manuscript is accepted. Subject to full method disclosure, you chair the review. We have here published in full, published partially, and referred to institutional biosafety committee. But the clean answer here is the governance follower is not choosing wrongly. It is having no forum, no criteria, and no record for making a choice at all. Biosafety governance is not a level. It is a 3 tiers that must interlock. If one fails, the whole chain breaks. The 3 tiers are global, including norms and obligation; continental, including harmonization and capacity; institutional, where safety actually happens. The chain breaks where the tiers do not talk, and the treaty obligation with no national law to domesticate it is just a paper. A national law with no institutional committee to apply is just ink. This is where we move from problems to solution. The Africa CDC Biosafety and Biosecurity Strategy of 2026 to 2030. It stands on 7 pillars: leadership and coordination by Africa CDC, multi-sectorial technical networking groups, regional and national, innovation and emerging technology, AI, synthetic biology, digital biosurveillance. Harmonizing and domesticating. We have legal and regulatory. We have regulation and certificate, professional training and certificator, and we have anchoring everything in national public health or national lab, reference lab. With the target of more than 60% of biosafety and biosecurity activity financed domestically by 2030. What we do next 5 years require country ownership, career pathway for biosafety workforce, govern innovation, real cross-border One Health cooperation. This is not a donor strategy. This is an African strategy for African resilience. Let me bring this down from strategy to my own work in Central Africa. We have yet one, genomic characterization of emerging viruses. Beyond the sequencer, you need culture by information trained bioinformatics and complete ethics and material transfer. Both are governance components, not technical ones. Regarding Binet 2, One Health and environmental surveillance. When you sample across human, animal, and environmental sectors, you multiply the number of institutions that must meet the same standard. In both cases, the technical work was fundable. The governance work was assumed to somebody else's line item, and that's exactly where the project failed. If you forget everything else from today, please take this away. Please take a screenshot of this 7 points of the institutional readiness checklist. I will close with this measure it or is not real. Documents do not make a system. Numbers do. 6 numbers separate a system from shelf of documents. If an institution cannot produce these 6 numbers today, it's lack of biosafety system, even if it has biosafety document. So, what now? 3 commitments in the next 19 days. Day 30, reconvene your biosafety committee. Day 60, complete a pathogen and data inventory. Day 90, adopt a dual-use screening checklist. None of the 3 require new equipment, but all 3 are what funder, partner, or regulation asks to see first. I will leave you with this. No governance, no growth. Thank you so much for this opportunity. Thank you.
Thank Thank you so much for that beautiful presentation. Please let's appreciate her. Let's show her some love. Thank you very much. We are glad to have you. And that was an insightful presentation. So we have come to the end of this webinar. We are grateful to our distinguished speakers for sharing their knowledge, their expertise and perspectives with us. And a big thank you to our participants for staying through to the end. Like we used to say, there is no Get Webinar series without our participants. So we are doing this because you are here. Thank you very, very much. So our speaker, Ms. Severance, James Diggins, Dr. Mary, Dr. Ketan, and Dr. David. Thank you for accepting our invitation and for contributing to this very important conversation. We are sorry we would not be able to take questions as we ought to because of our time. If you have questions, you can please direct them to getconsortium1@gmail.com. Direct your questions to getconsortium1@gmail.com. Thank you very much for joining. Also, we have a call for contributions for newsletter themed Building Resilience Against Infectious Disease Threats in Africa, Strengthening Preparedness, Response, and Regional Health Security. Thank you so much. If you consortium1@gmail.com, and the deadline for submission is September 11, 2026. Call for contributions. Thank you very much. Also, Dr. Mary, could you please stop sharing your screen? Thank you. Right now, I would like to take pictures with our partners. I would like to inform you that the webinar recording will be made available on our YouTube channel at Get Africa, our YouTube channel at Get Africa. So please do well to go to our YouTube to watch the recording of the YouTube. Also, certificates of participation will be made available to everyone who registered for the webinar. Certificates of participation will be made available to everyone who registered for the participants. We would like to request the indulgence of our speakers to switch on their camera so we can take a group picture, if you don't mind. I know a lot of our participants are off the call. Thank you. So can we have more speakers switch on their video? Okay. All right, 1 minute. We would enable participants to switch on their video now. That's why you can please switch on your video so we can take a group picture. Yeah, thank you. Let's as many as possible switch on their videos. Thank you very much for joining. We are glad to have you. On the count of, are we still switching it? On the count of 3. 1, 2, 3. Thank you very much. And do enjoy the rest of your day. Bye.
Oh, thank you so much. Bye-bye.
Thank you. Bye.